Welcome life science learners to another lesson. Trust that you guys are well and excited for our lesson today. In our lesson today, we're going to be focusing on revising genetics. It's an important section. There's lots of application of terms. There are problems that we need to solve. It's important that when trying to work on a section on genetics that we understand the terms. So as we get into the lesson, I'm going to spend some time looking at the important terms, give you an overview of the terms, and then try and see how we could use those terms to read through genetic problems and apply our understanding around solving these genetic problems. So whip out those books, grab a pen, and let's get busy. So as I mentioned, let's look at some core concepts and terms, and then we will look at some patterns of inheritance where we will discuss the difference between complete, incomplete, and codominance. So as we look at the terms, it's important that you make note of these terms. The concept of what is a locus. Again, guys, if we draw a chromosome or a chromatid, we know that often genes are indicated by colored areas or patterns on a chromosome. That specific position of a gene on a chromosome is referred to as its locus. So it comes from the word location. The next concept is alleles, and guys, remember that genes control the expression of characteristics, and these genes are inherited from the sperm and the egg, or we say from the mother and the father. So we get a set of two genes from both the parents. We know that these genes come in various different combinations when they inherited. So the possible alternative forms of these genes are referred to as alleles. So alleles can be represented for the height in plants, which you guys are familiar with when we looked at Mendel's experiment. These are the three possible alleles that we have. So we have these three illustrations as different types of alleles for a specific characteristic. The next term is homozygous. The word homozygous can be broken up into homo and zygous, meaning zygote. So this is an offspring that is produced by inheriting the same gene from both the parents. So for example, if we look at the characteristic of height, that is a homozygous allele, which means that this individual has inherited the same allele from both the parents, making this zygote a homozygous zygote. The term heterozygous refers to an individual having different. The word hetero refers to different. So essentially, this individual inherits contrasting genes from the parent for that specific characteristic. The next concept is dominant, and we will chat about the term recessive as well. So guys, it's important that we understand that the term dominant was coined when Mendel carried out his experiment. And when he did his experiment, he looked at separating out characteristics and then crossing them. And what he noticed that sometimes in a cross, he noticed that when a characteristic crossed with another plant produced one characteristic in the offspring, he referred to that as being a dominant trait, a trait that prevents the recessive trait from expressing itself. So that means we've got to understand what the word recessive means. And essentially the word recessive points to a trait or characteristic that is expressed only in the absence of the dominant trait. So when the dominant trait is not present, example in a homozygous combination, we refer to that being the recessive trait expressed in the absence of the dominant trait. Again, the word homozygous dominant would refer to an allele that has, again, the same allele present from both the sperm and the egg. The word heterozygous allele points to a zygote that has got contrasting alleles for the same characteristic. So that would be a different allele from each parent, i.e. sperm or egg, that come together. And finally, the term homozygous recessive would again point to an individual that is recessive and having the same gene, the word homo meaning having the same gene inherited from the both parents. Homo, same, zygote, again, same alleles from the parent. Okay. We quickly wrap this section up with a few more terms such as genotype and phenotype. Again, this points to The genotype is the genetic makeup, which is often expressed using alleles. So when we illustrate these, we refer to these as the genotype of the individual. Phenotype is a description of the physical appearance of an individual. So when we talk about the plant being tall or an individual being right-handed, we refer to that as being a description of the physical appearance of a characteristic. Guys, you would also remember that when we study genetic crosses, a cross can be done for a single characteristic, so we call that a monohybrid cross, or we could study how two characteristics, di meaning two, are inherited at the same time. So these we will discuss again in our lesson. We do know that in a genetic cross, when parents are crossed, we know that they produce an F1 generation, and the F1 generation points to the offsprings produced in a cross. So the filial generation would be the offsprings produced when parents are crossed. And finally, the concept of a Punnett square, again, is a systematic tool that we use to find out what are the possibilities when we cross individuals. So again, it's an important tool that you use in a genetic diagram to illustrate the possibilities of offsprings that are created. Guys, we're going to look at the concept of complete dominance in our lesson, where one characteristic prevents another characteristic coded for by that gene from expressing itself. So that's one pattern of inheritance. The other pattern of inheritance we refer to as co-dominance. Again, this is an example where a type of dominance where both alleles are expressed fully. An example of this are human blood groups, where you have A blood group, B blood group coming together and being represented as an AB. So it's a combination of both. So it's a co-dominance pattern of inheritance. And finally, the third pattern of inheritance that you need to remember is incomplete dominance. So this essentially is when the dominant gene allele is not able to completely dominate over the recessive gene allele. So this results in a mix of the two genes, which creates what we call a new phenotype. So the example that we often refer to is in certain species of plants, when a plant that is homozygous red is crossed with a plant that is homozygous white, they produce an offspring that is pink. And pink means now we have an incomplete dominance of red over white, which results in a new phenotype. So remember that in incomplete dominance, you end up with a new phenotype in the offspring. Guys, we've looked at the important terms. We've had an overview of the different patterns of inheritance. I trust that you guys have recollected those important terms. Let's go into a break, and when we get back, we're going to apply some of these terms into problem solving. So have a short break, and I'll see you in a bit. Welcome back, life science learners. We were looking at terms and we're revising genetics. Now let's apply some of that understanding to a few questions, and then we'll get on to some detailed questions on how we apply the concepts of patterns of inheritance and how genetic diagrams are laid out and applying those terms in the context of different types of questions. So before we went into the break, we had looked at the terms. Let's see if we can answer some questions based on descriptions. So the first question was, if we go back to this, was, let's see. So we've got to provide the correct biological term for the following. A, different forms of a gene located at the same position on homologous chromosomes. So again, it's a lot that we need to take in, but we've got to read every aspect of that. So different forms of a gene located at the same position on a homologous chromosome, again, refers to the alternative forms, and that concept or term is allele. So that points to alleles. The next term or description that we need to find out would be B, the appearance of an organism due to its genetic makeup. Guys, remember that it's the appearance of an organism due to its genetic makeup. So when we look at the appearance of an organism due to its genetic makeup, we refer to that as the phenotype. So when we talk about the appearance of an individual due to its genetic makeup, we say that that is the physical expression of a characteristic. So when we describe an individual being tall or left-handed, we say that that's the physical expression of the genes. A few more and then we'll get into some detailed application. An organism with unlike alleles for a particular characteristic. Guys, this is again a fancy way of saying different, unlike. So when we talk of unlike alleles, we're meaning not the same. And if you recollect, not the same would refer to something that has unlike alleles. Or if we talk about right-handed and left-handed, these individuals, we refer to them as being heterozygous individuals. So the term heterozygous again refers to organisms having different or unlike alleles. So another word for unlike would be different alleles for the same or for a particular characteristic. The next concept, D, an allele that expresses itself in the phenotype. We know that an allele that expresses itself in the phenotype often is the dominant allele. So when we talk about dominance, we know that it is that characteristic that is expressed in the phenotype, regardless of whether it is in a homozygous or in a heterozygous combination of alleles. And finally, the genetic makeup of an organism. If we talk about the genetic makeup, we're talking about the genotype of an individual. So that's a wrap of basic application of some of the terms that we've just looked at. Let's take this to the next level and try and apply this to questions that often spell out a series of crosses between parents and give you a result of that, and then requires us to be able to use our understanding of terms to figure out the genotype ratios, the phenotype ratios, or even sometimes the possible offsprings that may be produced. So I encourage you to try and work these with me, and as I get through them, for you to test or to check your answers. Right, so question 3. In horses, black coat color, and I note that I am highlighting the important concepts in a genetic diagram, is dominant over white. So we know that black, again I enjoy doing this in that I make sure that I write down clearly what the dominant and the recessive is just next to my question, and that helps me to clear my head around not being confused as to what is dominant and recessive. Let's read further. A white mare mates twice with the same black stallion. She produces a white foal on the first occasion and a black foal on the second occasion. Use the letters B capital and B small as indicated above and write down the genotypes of the following. Now guys, it's important that when we have a question like this, that we're able to unpack the question and make sure that we interpret the question. This is often about reading the question carefully and understanding which individuals and their genotypes that are involved in this. So there are 2 crosses, and I'm going to try and write that down. So it's a white mare. So I'm just going to write that down so that a white mare is the first cross that is crossed with a black stallion, okay? And that's crossed with a black stallion. And that's the first cross, and that happens the same time again. She produces a white foal on the first occasion. So you'll find that I'm just going to go up here. The first occasion produces a white foal. That's on the F1 on the first generation. And then again, another process happens and they produce a black foal. Now guys, it's important that we understand that here we need to understand what the possible genotypes for black are. So black has 2 possible genotypes because it's dominant. It could be homozygous dominant or it could be heterozygous. Whereas white can only be the homozygous recessive combination. So it's important that we have this understanding before we get into trying to solve this. So when we talk of white or when we refer to white, it's important that white can only be recognized as that. When we refer to the male being black, it could be two possibilities, guys. It could be either homozygous or heterozygous. How do we determine that this individual is homozygous or heterozygous? We've got to look at the next generation that is produced in that cross. And remember that it produced a white foal in the first cross, and the second time they produced an offspring, it was a black foal. Now guys, it's important that we can use any one of these individuals to figure out what the genotype of the male was. So let's write this first cross down. So that was the white offspring, and the second time they produced a black offspring, and that black offspring could be either that or that, and we're not too sure about that as well. But this is the clue, guys. Here's the clue. And so this tells us that this individual who is white has to inherit a recessive allele, meaning a small b, from both the parents. So if we reflect on this, it means that, let's look at this, it means that this small b, comes from the mother, and the other small b has to come from the father. And in this case, it cannot be this individual, or else you will not get white foal in the offspring. So it means that just purely by looking at this individual, we are able to identify that the father, in this case who is black, has to be heterozygous. And so that is the initial unpacking that you need to do in this question. And this helps you to now read into the questions and have a better understanding of how to answer the questions that come. So let's look at this. The mare and the stallion. So guys, as we just worked out, the mare is the female, and she is, again, if I were to describe her physical characteristics, she is white, and white in this case can only be homozygous recessive, so small b, small b. When we look at the stallion, we just proved that, yes, we know that he is black, but we realize that there are two possibilities of black, and we, purely by looking at this offspring, we realize that the male has to be heterozygous, and so his genotype is going to be capital B, small b, in other words, heterozygous. Let's move on to more questions based on this. As I mentioned, here we looked at the answer for that, and we said that the mare is that crossed with the stallion who has to be that, because if he's not, they will not be able to produce a white foal that has two recessive alleles. And that's the explanation to why the male is heterozygous. Now let's determine the genotypes of the first and second foal. Now if you go back to the question, the first foal, if we look at the parents, and I'm going to go back to the parents we said was white crossed with a black who is heterozygous, and they produced a white foal on the first cross, and they produced a black foal on the second cross. We know that white can only be that, and we know that black could be either. But knowing that the mother is homozygous recessive, it would mean that she has to contribute the recessive alleles to each of the offspring. So when we look at the male, he inherits, or the female contributes to one recessive allele from the mother, and the other has to be the dominant allele from the father. And hence it cannot be that, and can only be the heterozygous combination. So the answers to that would be the first white foal would be that, and the second black foal would be that. And I trust that you guys are now understanding the reasoning behind how we get these answers. Right, so guys, let's attempt another question. These are interesting questions because they test you on your understanding of the concepts, and simply read through the questions well. I'm going to do that, and I want you to follow my method of working through this particular problem. In guinea pigs, the gene for black coat is dominant to the gene for white coat. So we're going to do this as I do always, the black coat is dominant over the gene for white coat. So white again is the recessive allele. Okay. 2 heterozygous black guinea pigs are crossed. So again, the term heterozygous would be that are crossed. So 2 heterozygous black guinea pigs are crossed. Now that's very quickly establishing the genotypes of the parents. Let's read the first question. By means of a genetic diagram or a diagram, show the genotypic results that would be expected in the F1 generation. And this is key, guys. It's a 6-mark question. Guys, it's important that we realize that in this question, when it's a 6-mark question, that you have to illustrate the entire genetic diagram from the parents using a Punnett square right till we get to the F1 generation, showing us the genotypes and phenotypes of the F1 generation. Let's try and do that as we wrap this segment up. So here's a template that I use for the genetic diagram. And this genetic diagram again has to be laid out systematically, as you would see. And I'm going to go back to very quickly taking note of our possible genotypes that we need to use. So these are our genotypes of the parents and 2 heterozygous black guinea fowls. So we'll start off with the phenotypes, and they are black, and we know that both are heterozygous, so I'm going to keep that at the back of my mind, and I'm going to make a note of that they both are heterozygous. And that's important, as we established that the genotypes of the parents have to be heterozygous, and that is clearly indicated. We then need to show the gametes that are produced, and it's good to often use the gametes by indicating that they are circled, and that helps to differentiate that these are gametes and not just the homozygous genotypes written together. The next process is fertilization, and as I mentioned earlier on, we use this illustration called a Punnett square, and you're familiar with this, and where we indicate the gametes from a parent here, and on the other row we indicate that. This is showing you the possibilities that might occur, and we derive this where we cross that individual with that, and so on. So that will be the next individual. This would be a cross between the B here and that small b there. And finally, this is a cross of the small recessive b's. And this then gives us the possibilities in the F1 genotype. And so here we have, I'll start off with the homozygous dominant, which is one of that. We now have 2 of these heterozygous individuals, and I'll circle that out there for us. So that's 2 capital B small b's. And finally, we've got just the single individual here with a homozygous recessive. So we refer to this as the genotypic ratio, where we have 3 possibilities produced. Finally, we've got to end up with the phenotype ratio, and we've got to now describe the physical appearance of the offsprings. And we know that that's black and that's black, so collectively they are 1 plus 2, and I'll call that 3 black, to one of these, which in this case is the white individual. So guys, this is a genetic diagram. I'm going to wrap this segment up by showing you the solutions and a quick illustration of where the mark allocation for this particular question is. As we note, there's an important mark given for showing the genotypes of the parents, and that's black fur on both sides. We then need to illustrate the genotypes of these, which are illustrated there, which I've got correct. We show the possible gametes, which I indicated earlier on using these letters, and I circle them. And then we show the Punnett square with the possibilities that develop. And as you would see, we've got this ratio at the end, which is a 25 and a 75% ratio, indicating the black to white possibilities in the offspring. Welcome, life science learners, back from the break. Trust that you're excited. We're revising genetics. We've looked at how to solve some genetic problems using a genetic diagram. Let's look at some pedigree diagrams and look at how we interpret these in this segment. Let me remind you that a pedigree diagram is essentially a family tree that shows you how a specific characteristic is passed on from one generation to the next. And these are important tools in understanding what are the possibilities of the next generation inheriting a characteristic when two individuals are crossed. So let's look at some of these genetic diagrams illustrated as what we call pedigree charts. Let's get straight into that concept. So again, a pedigree chart, as I mentioned, is a chart that shows the presence or absence of a trait within a family across generations. It's important that let's understand what these symbols represent. So often we have a key that says that circles represent the females in a population, the squares represent the males. When we have a line connecting the individuals, we say that that is a marriage line, or in the context of animal, it's a cross that happens. So that would be a line that indicates individuals being crossed, and a vertical line connecting two individuals together that projects down indicates the possible offsprings that are produced. This line that's horizontal and that connects individuals, so I often refer to this as the T, the upside down T, and that indicates the siblings that are produced or the different offsprings. And when we illustrate numbers along this side of the diagram or on either the left and right hand side, these indicate the generation numbers. So this diagram can be interpreted as a cross between a male and a female that produced two offsprings, and these offsprings were a male offspring and a female offspring. There were two generations, and that's how we interpret a pedigree chart. However, it's not that simple. That was purely an indication of the symbols. Now let's try and understand and apply that in the context of some genetic problems. So let's read through this. Study the family tree which shows the inheritance of a type of earlobes over 4 generations of a family. In humans, free earlobes is a dominant trait over attached earlobes. Now guys, I'm not too sure if you've ever noticed this, but when we look at our pinna, there are possibly 2 ways in which the pinna might be attached. And if you look at individuals, maybe someone sitting around you, you will notice that in some individuals the pinna is free. In mine, if you look closer, it's slightly free. In other individuals, you'll find that the pinna is attached to the kind of the cheekbone here. And so we say that that is fused. So there is a difference between a fused and a free pinna. So again, here we're looking at fused earlobes and free earlobes, noticing that the dominant is the free, so we say that capital F is for free, and the fused in this case would be the small f. And guys, again, it is important that if you are used to writing your alphabets, the capitals and the small letters similar, try and differentiate them clearly by indicating larger meaning dominant alphabets and smaller alphabets meaning the recessive alphabet, especially with letters like S, where some of us tend to write our S's similar. And so if you want to differentiate between a dominant and a recessive, try and be very meticulous in that description. So even when we use the letter T, some of us tend to write a small t using that, and that's quite clear that it is recessive. So be careful as to how you illustrate your F's, and I've indicated my F clearly with a little cursive or a clearly indicated smaller f. So here's the diagram, and again, let's understand that in a pedigree diagram, it can be very confusing. You've got to first establish what are the genotypes of the individuals. And so I often help my learners remember that there are many clues in a pedigree chart. We've got to find the clues and use these clues to work either to the next generation or backwards a generation. So again, I want to remind us that F is free, and that's the dominant, and small f is fused, and that's the recessive trait. If we were to illustrate the possible genotypes, there are 3 possible genotypes that we always look at, and that's the only genotype for fused. And this means in the context of this chart, let's look at the key. So males with attached earlobes are unshaded. Males with free earlobes are shaded. So this means that free would be capital, capital, or capital small. So these are the possibilities. We know that the free earlobes have to be these. And so when we look at this illustration, it's important that when we go through this, we identify the free earlobes as being the dominant trait, and we allocate these individuals a capital letter F. And I often do that quickly before I get into unpacking the possibilities in this cross. So as you would see, I've allocated all the individuals having the dominant trait a capital letter. What's also important to realize is that the attached earlobes are the recessive trait, and these can only be the individuals that have a homozygous recessive combination. And so guys, this means that these individuals have to have two recessive alleles. And so all of these individuals that are unshaded in this case have two recessive alleles. And I'm going to very quickly do that and illustrate that for you. Guys, I know that this is confusing you because conventionally in textbooks, often individuals that are affected, we tend to show them with the shade. Unfortunately, because of the way we present our slides, we've got to change the color of individuals, and hence you're seeing a color that normally is non-shaded, shaded in this. Again, it's a principle that we need to recognize. So as you look at this diagram, you can clearly see that I've gone through and I've illustrated these individuals that have genotypes that are homozygous recessive, as well as individuals that are clearly dominant. So let's unpack this question by going into the rest of it. So the first part of this question is, how many members of the family have free earlobes? Guys, if we go and look at the chart that shows you free earlobes, it's all individuals that are shaded. And so essentially we've got to go and count the individuals. So it's 1, 2, 3, 4, 5, 6, 7, 8. So clearly the key helps us to determine which are the individuals that are free, and we have established that these are 8 individuals that have earlobes that are free. The next question would be, what proportion of offspring in the 4th generation are females with attached earlobes? Now let's go and count the generations. So that's the first generation, the second generation, the third generation, and the 4th generation. So we've got to determine in the 4th generation what is the proportion of females with attached earlobes. So if we look at the 4th generation, we've got 1 female here, we've got another female. So we've got 2 females, and of the 2, one of them has what we refer to as the attached earlobe. So this individual here has the attached earlobe. So simply put into a ratio, of the 2, there's 1 out of the 2. So we can say that there's 50% of them that have a attached earlobe. So guys, that's a very quick illustration of a pedigree chart and how we interpret that. I think we're going to have another opportunity to go through and unpack more pedigree charts, but you need a break. Let's have a little comfort break, and I'll see you in a bit. Welcome back, life science learners. Let's continue with our revision lesson on genetics. We were looking at pedigree charts before we went into the break. I know that these can be very confusing and often require a further detailed explanation. Let's take a few more questions on this and then look at another pattern of inheritance where we refer to as sex-linked inheritance. I wasn't complete with that question before we went into the break. I'd like to go back to that and unpack some of that when we look at how pedigree diagrams are interpreted. So again, I'm going to go back to this, And I'm going to refer to this chart. And the question that we're going to get onto is it possible for individuals C and D to have a child with free earlobes? So guys, when we look at individuals C and D, if we remember, I illustrated this earlier on that again, the free earlobes is the dominant trait. So we refer to them as being either that or that. So homozygous or heterozygous. And the individuals that are free will have to have the recessive alleles. So essentially, these individuals have to have 2 recessive alleles. So knowing that these individuals are homozygous recessive, let's try and unpack the next part of the question. Is it possible for individuals C and D to have a child with free earlobes? So essentially, if we have these individuals that are crossed, no matter what happens in a genetic diagram or a pedigree or Punnett square, guys, we know that all of these individuals that are produced will always be homozygous recessive, as I've indicated in this Punnett square. So the answer would be, is it possible? The answer is no. And I know that it's not simple enough to be able to settle for no. Let's try and understand why you say no. And essentially, my explanation to this would be, guys, if you've got parents that are homozygous for a particular characteristic, and both these individuals we refer to as pure breeding, when we cross those individuals, the only possibility that we have in the next generation is the same characteristic appearing. And so in this case, when these parents are crossed, there is no other allele but the recessive alleles that can be inherited. And so essentially we would say that since C and D have attached earlobes, they have only the recessive genes, and as I mentioned, these are the recessive genes, and can therefore have no dominant gene or genes for free earlobes to be passed on to their offsprings. So it's literally impossible for that to happen. Okay, and guys, as I mentioned earlier on, the key to working out many pedigree diagrams is to identify what I refer to as the individuals that are affected or homozygous recessive, because you've got to know that when an individual has these recessive alleles, it means that both the parents have to have the recessive allele. So when we look at this individual here, which is individual F, and if we allocate that individual the recessive alleles, it's important that we recognize this fact, as that during independent assortment, each of these alleles come from the parent. And we know that this means that individual F would have inherited a recessive allele from the mother and a recessive allele from the father. And that is crucial to individual F inheriting 2 recessive alleles. And then that should help us to determine what the father is. And because he has free earlobes, which is the dominant trait, he has to have the heterozygous combination. So again, guys, the golden rule is to look for individuals that are affected with the condition. Again, individuals that are affected are homozygous, as I mentioned here. And use the rationale of asking yourself, where did they get those 2 recessive alleles from? And that means they would have got it from their parents, making their parents at least heterozygous. They could be homozygous, recessive, they could be heterozygous, but they cannot be homozygous dominant. So you will not get an individual that has that producing an offspring that has that. So that's the key. And again, you can use that same rationale to infer, like we would in this example, that this individual, which is the female, can only give all the offsprings the recessive allele, meaning that all of these offsprings have to inherit a small f from the mother. And that clearly is because she has 2 recessive alleles, and all that she can pass down to the next generation is the recessive allele. And that is the key to working with pedigree charts, is to first establish the individuals that are homozygous recessive, and then use them again to determine what they pass on to the next generation, or to determine what they have inherited from each of their parents. And that's the clue that I would like to leave you with as we move on to the next bit, which is looking at sex-linked inheritance. So guys, when we talk of sex-linked inheritance, again, it's important that we recognize that gender is determined by specific chromosomes. And we've looked at this in the past, where we know that individuals have 2 chromosomes which indicate the gender. The X and X chromosomes indicate that the individual is female, and the presence of an X and Y chromosome indicates that the individual is male. It's important that we recognize that on these chromosomes that they are genes, and these genes are inherited in a specific pattern, and we refer to that as the sex-linked inheritance. Cool. So we spoke about the X and Y being the sex chromosomes. The Y chromosome is much shorter than the X chromosome, which essentially means that it contains fewer genes. All males have a Y chromosome, and that Y chromosome is significantly smaller. On the other hand, the X chromosome is a longer and contains significantly more genes than the Y chromosome. Here's an electron micrograph indicating a comparative size between what we call the X chromosome and the Y chromosome. And essentially, the concept of a Y chromosome is derived from the appearance of this, which appears much shorter based on the position of the centromere. But we'll chat about that at some other point. So this is essentially showing you how different the X chromosome is and the number of genes that it contains, whereas the Y chromosome is significantly shorter, having fewer genes on them. When assigning alleles for sex-linked traits, the convention is to write the allele as a superscript for the sex on the sex chromosome. We're going to unpack that by referring to some characteristics that are sex-linked. It's important that you take note of a few of these genetic disorders that are sex-linked. It does help when you know these, because when you do read this in the context of an exam or test, you know immediately that this is a pattern that needs to be sex-linked. So hemophilia is one example. Color blindness. Cystic fibrosis is another example. So these are 3 common examples of characteristics where they are found on the X chromosome. And they follow a specific pattern, and we've got to recognize that in our next slide. So the example we're referring to is hemophilia. And we tend to use, again, the gender as well as the allele as a superscript. So essentially, you've got to indicate the X, dominant and recessive alleles using that notation. And again, we have the Y chromosome in males that does not have the allele. So remember that this is an X-linked characteristic. Often we get learners indicating on the Y chromosome the presence of the allele, and that's wrong. Again, it's an X-linked characteristic. It can only be found in the X chromosome and not on the Y chromosome. So that would be an incorrect way of representing sex-linked characteristics. So let's get straight into trying to work through a problem. Hemophilia is a sex-linked disease caused by the presence of a recessive allele indicated using that. A normal father, and often like to use the word unaffected. Again, the context of normal meaning hasn't been affected, unaffected. And a heterozygous mother have children. So guys, the concept here is that the parents, firstly, the male is unaffected, and that is a cross with a heterozygous female. So the female is heterozygous. And I think when you read through a question like this, it's important that we recognize early on in the question the genotypes and the phenotypes of the parents. When we refer to as a male being unaffected, we need to say that it's a male that is unaffected. Essentially, because it's sex-linked, we need to indicate the gender of the individual. And that's key to solving genetic problems using sex-linked inheritance. Again, because it is a male, we had to indicate his gender. So that's the first step. Crossed with a female, again, indicate the gender. Now we determine what the alleles involved in this. Again, the male is unaffected. There are 2 possibilities of males, either being affected or unaffected. So again, knowing that hemophilia is a recessive condition, it means that this male who is not affected has the dominant allele, because we noted that that's the notation for the recessive allele. It's important to recognize that the female is heterozygous, meaning that she has the dominant and the recessive allele. Again, so I've got to allocate a heterozygous combination to that. And that's fundamental to getting the rest of the problem correct. So now we've established the genotypes of the parents. So let's read the question. Represent a genetic cross to determine the possible genotypes and phenotypes of the children of the parents mentioned above. And that's a 6-mark question. Guys, again, this is a genetic cross using a genetic diagram. Let's refer back to our template and let's try and unpack that. So again, I'm going to very quickly illustrate that. Again, we had the phenotype of the parents, so it was an unaffected male. And it's important that I indicate the gender as well. Crossed with a heterozygous female. And this is the important bit now, guys. The genotypes. So the male is XY crossed with a female that's XX. At this point, we need to indicate the allele. So it's an unaffected male, so capital H, heterozygous female, small capital H, small h. Note that I've clearly indicated the difference between my capital letters and the smaller letters. It's important that we now form gametes and we separate them out. I often encourage my learners to circle them to indicate that these are standalone gametes that are haploid. So essentially what we're doing is we're separating out the alleles into these gametes. The next bit requires that we use the Punnett square to illustrate the possibilities that might occur in a cross of this nature. We now have the male and female gametes illustrated on either row and column. The next bit's quite easy, but you need to remember that it's important that we first cross the gender, and I'll illustrate that. So let's cross the gender, and let's do that. When we did the first cross between these two, we note that there is a dominant allele there from that parent here, and a dominant allele from that parent there. Likewise, here we get the Y from this parent, and we get the H dominant from that. And so I'm going to quickly wrap this up for us as we conclude this problem. And I'm going to get through to that individual who is now going to be recessive. Looks a bit messy, but we can quickly illustrate that as being one individual, female, who has that. So I've ticked that off. Notice the next female is this individual here who is heterozygous, which I've kind of now ticked off. And this helps me to be able to ensure that every individual in that Punnett square has been presented in my genotype and phenotype ratios. So what I've quickly illustrated here is the possibilities of having individuals. And finally, as we wrap this segment up, we've got to write down the phenotype ratio of the individuals. So we've got to indicate the gender, guys. So that's going to be a female that is unaffected because she has both dominant alleles. So that's an unaffected female. The next is a female, but if you look at her genotype, she's got the dominant allele and the recessive allele, making her what we call a carrier. So she's not affected by the condition, but has the recessive allele and may pass it on to the next generation. And finally, we've got the males, which we have to indicate as males, and this individual has the dominant allele being unaffected. And the last male, again, being a male that has the recessive allele, so that's going to be an affected individual. And guys, there are many ways that these questions can be asked. One could be determine the possibilities of having an offspring that is affected. And we've just established that there is one male of the 4 that could be affected, a 25% chance. Guys, you've been a fantastic audience today. Trust that you've enjoyed the interaction, that you can go and work with some of these problems confidently. Wish you well. Have a lekker day. See you soon.